let's talk now a little bit about ATP and enzymes. If you think of a cell as a small, busy city, there are all kinds of proteins that move substances into and out of the cell. There are motor proteins that carry cargo along microtubule tracks, and there are enzymes that busily break down and build macromolecules. Even processes that aren't energetically favorable, those that are exergonic or spontaneous, will occur only if there's energy available to power them. If the energy runs out, the reactions stop and the cell dies. These energetically unfavorable reactions are often paid for by what we call by being coupled with energetically favorable reactions that release energy. Often the payment reaction involves ATP, in particularly the hydrolysis or the breakdown of ATP. That's the energetically favored reaction that powers those energetically unfavored reactions. ATP is a small molecule. It's a nucleic acid. As you can see, it's composed of a sugar, ribose, just like RNA, a nitrogenous base, adenine, and 3 phosphate groups. It's unstable. These 3 negative phosphate groups repel each other. This last bond right here is particularly unstable, fairly easy to break, and when it does break, as we can see in this reaction, when it's hydrolyzed, this is ATP hydrolysis, some of the energy stored in that bond is released. Like most reactions, it's reversible. So cell respiration essentially makes this reaction go in reverse. Cell respiration takes ADP, energy from food, and uses it to make ATP. So here's what we call the ATP cycle. So let's think of ADP and P as a dead battery. So energy is put in to those from food during cell respiration, water's released, and we get a charged battery. We get ATP. So this is what's happening in cell respiration. That ATP can then be hydrolyzed, and you see the water coming in as a reactant, and that the hydrolysis is going to release energy to power any of the body's reactions that need energy. The delta G, the change in free energy for the hydrolysis of ATP, is about negative 14 kilocalories per mole. That means the reaction is extremely spontaneous. And it means that ATP is very, very reactive. It's very, very difficult for the body to store it and transport it. And that's why plants actually use ATP to make glucose. Glucose and other sugars are more stable and more storable. Eventually, they're going to use that glucose to remake ATP, but they need to make the sugar so that they can store and they can transport the energy. The energy from ATP is often used, as we said earlier, to power endergonic reactions. And typically what that means is that these two reactions happen together. They happen in a pair. So the hydrolysis of ATP is going to release energy. The endergonic reaction is going to absorb that energy. So as it says here, during coupling, an energetically favorable spontaneous reaction, like ATP hydrolysis, is linked with an energetically unfavorable endergonic reaction. The linking often happens through some kind of shared intermediate, but the key idea here is this. When two reactions are coupled, they can be added together to give an overall reaction. The delta G of this overall reaction will be the sum of the delta G values of the individual reactions. And as long as that overall sum of delta G is negative, both reactions can take place, the spontaneous one and the non-spontaneous one. So that's coupling. When a endergonic reaction like ATP hydrolysis, let me say that again, when an exergonic reaction like ATP hydrolysis is coupled with an endergonic reaction in order to provide energy to make that endergonic reaction happen. So let's talk about enzymes. Enzymes are proteins which catalyze chemical reactions. They do this by lowering the activation energy required for a reaction to happen. Enzymes act on substances called substrates, and the substrates tend to bind to an area on the enzyme known as the active site. And we can see that here. Here's the enzyme. Here's the active site. Notice it has a specific shape. It also has a specific charge. The substrate will fit in there, kind of like a key fits into a lock. So sometimes we'll say this is an enzyme plus a substrate. Once they get together, we have what's called the enzyme substrate complex. Typically during this time, the enzyme will go through what's called a conformational change or an induced fit. And it will act upon that substrate to either break it down or put two different substrates together possibly. And what we end up with is the enzyme is unchanged, but we end up also with products. So this means that the enzymes aren't changed by the reaction. They can be used over and over and over again. And they are very specific because only one substrate will fit into that active site. Some enzymes catalyze anabolic reactions, so some do build up reactions, while others catalyze catabolic or breakdown reactions. We already said that the enzymes themselves are not used up or changed by the reaction. They can be used over and over again. Another important thing to know is each enzyme works best at a very specific optimal temperature. For human enzymes, it's usually body temperature, 98.7 or 37 degrees Celsius. They also work best at a very specific pH. If either of those is changed and moved away from optimal conditions, the enzymes can begin to denature. That means the proteins that the enzymes are made out of change shape, and therefore they no longer work. So here are some factors that can affect the rate of enzyme catalyzed reaction. Concentration of the enzyme. Higher concentration typically means faster rate. Concentration of the substrate. Typically a higher concentration of the substrate is going to mean a faster rate of the reaction. Now that's up until a certain point when all of the enzymes are full. Past that point, it's not going to help. Higher temperature typically means faster rate. Now that's up to the point where the enzymes start to denature. If you go past that optimal temperature, the enzymes will start to change shape and won't work. pH. So if the pH is moved away from the optimal pH, either up or down, the rate of the reaction will slow. Also salinity or saltiness. If the salt levels are changed, either too high or too low, the rate of the enzyme catalyzed reactions will slow as well. So let's also mention enzyme inhibitors. Inhibitors are substances that attach to and inhibit or prevent enzymes from working. We usually talk about two kinds of inhibitors. First, let's mention the competitive ones. These are molecules that compete with the substrate for the active site. They may have a shape that's very similar to that of the substrate. They slow down the rate of the reaction because they prevent the substrate from binding to the active site. There's also non-competitive inhibitors. These attach to a spot on the enzyme other than the active site. But the net effect is they cause the shape of the active site to change so the substrate can't fit. These molecules can cause the reaction to stop permanently. So if an inhibitor is competitive, it decreases the reaction rate when there's not much substrate, but can be outcompeted if there's a whole lot of substrate because they're competing for the same place. If an inhibitor is non-competitive, the enzyme catalyzed reaction will never reach its normal maximum rate, even with a lot of substrate, because it's not competing with the substrate for a spot. The spot that it binds is open, it will bind there, and that prevents other substrates from binding to the active site. So here we see normal binding. Substrate has a complementary shape to the active site. They fit together, and we'll get an enzyme substrate complex. Here we see a competitive inhibitor. That thing has bound to the active site, and it has blocked the substrate from binding there. Here we see non-competitive inhibition. So the non-competitive inhibitor bound to a site, changed the shape of the active site, and the substrate can't fit in there anymore. So let's also talk about what's called feedback inhibition or allosteric inhibition. This is the temporary deactivation of an enzyme or a metabolic pathway brought about by an elevation of an end product of that pathway. It's essentially a type of non-competitive inhibition. We might also hear it called allosteric inhibition. Typically, the product of the pathway, of the metabolic pathway, binds to one of the early enzymes in the pathway. And it binds to not the active site typically, but it binds to a spot on the enzyme called the allosteric site or the feedback site. Either way, that changes the shape of the active site and temporarily inhibits the enzyme from working. The process is dependent on the concentration of the end product. When the concentration is high, the pathway is inhibited. When the concentration is low, the pathway resumes its normal work and begins to produce the end product. Feedback inhibition is essentially negative feedback. It's an important way for cells to conserve resources. So here we sort of see it diagrammed. Here's a reactant. So this is a metabolic pathway. We've got 5 enzymes involved and 5 steps. Ultimately, this metabolic pathway is trying to make substance F. When the concentration of substance F gets high, some of that substance F binds to the allosteric site on enzyme number 1. Well, that changes the shape of the active site. That means enzyme 1 no longer works. Well, because enzyme 1 no longer works, enzyme 2 can't work, because the product of the enzyme 1 reaction is the reactant for the enzyme 2 reaction. Well, that also means that 3 can't work, because the product of the enzyme 2 reaction is the reactant for 3. And that continues for all these enzymes. So by inhibiting the first enzyme, essentially the whole pathway is inhibited. And it stays that way until the concentration of this end product starts to drop and it becomes low. When the concentration becomes low, the molecule that's bound to this allosteric site is used up also. The active site of enzyme 1 goes back to normal. It starts to make substance B again, which means enzyme 2 can work. It starts to make substance C again, which means enzyme 3 can work, and the whole pathway turns back on. So feedback inhibition is about carrying out a process when you need an end product, when the concentration is low, and then stopping that process when you have enough of it and the concentration is high. It's about conservation of resources. All right, we'll also mention enzyme activators. These are molecules or sometimes ions that enhance an enzyme's ability to work. So they're sort of the opposite of inhibitors. We'll talk about 2 kinds of activators. Cofactors are typically inorganic, not carbon-based. Usually they're metal minerals like magnesium, iron, or zinc. They attach to specific spots on enzymes and essentially activate those enzymes and make them work. Coenzymes are similar in function, but different in structure. Coenzymes are usually large organic molecules, so large carbon-based molecules, oftentimes referred to as vitamins. Again, they attach to certain spots on specific enzymes and activate those enzymes. Vitamin and mineral deficiencies are dangerous because without them, certain enzymes don't work. They never get activated. All right, that's it for energy and enzymes.